Good day. Welcome to today's lecture series on lipids. In our previous lectures, we look at the chemistry and structure of fatty acids. We also look at the chemistry and structure of triacylglycerol. Today we'll be focusing on the structures and chemistry of glycerol phospholipids. Now let's look at phospholipids or glycerol phospholipids. A 1,2-diacylglycerol that has a phosphate group esterified at the carbon atom 3 of the glycerol backbone is a glycerol phospholipid, also known as the phospholipids. Now, if you look at the structure here, you will find that we have the So we have, this is carbon 1, carbon 2, carbon 3. So we have the esterification here, which makes it a monoacyl. We have another esterification which makes it a diacyl. Then the phosphate group that makes it a phospholipid. So the name is derived from the glycerol and the phospholipid. That's why it is called glycerol phospholipids. Now these lipids form one of the largest and most important class of natural lipids. They are essential component of the cell membrane and are found in small concentration in other part of the cell. When we look at the membrane leaflet, we find out that we have the membrane bilayer. This bilayer is made up of these phospholipids. And we have examples of these phospholipids, which I'm going to discuss later in this lecture. Now, it should also be noted that all glycerol phospholipids are members of a broader class of lipids known as phospholipids, which we've actually talked earlier. So, this is a phosphatidic acid. What we have here is a phosphatidic acid. You can see the esterification, first esterification. You can see the second esterification. Then this is the esterification of the phosphate group. Now, in naming phospholipids, there are things we consider, which is the stereospecific numbering system. How do we number the glycerol phospholipids? You can see, for example, this is a glycerol having 3 carbon atoms, 1, 2, 3. So you can see that the glycerol has 1 hydrogen here, hydroxy group here. Then let's say an hydroxy group, usually the methyl group. We can actually call alcohol group, but it's attached to a carbon. You also have another one here. You find out that this carbon center is a prochiral center. Why am I saying it's a prochiral center? Because we have 1 hydrogen, this is a different functional group, 1, 1, 2, then the functional group 3 and the functional group of this one is the same. That is why it is called a prochiral, meaning that the substitution of one of this one or this one will make the center a chiral center, and that chiral center makes it to have a mirror image, or will actually make the molecule that exists in the D or the L form of the molecule. So you can see here that the replacement of 1 hydrogen with a deuterium atom, which is hydrogen 2, is actually changing the configuration to S configuration, which is SRS configuration system, which you use in prochirality naming. So you can see in this structure, we have the pro-S position, we have the pro-R position. Here you can see that the functional group here is different from the functional group here, which is also different from here and here. So this center is a chiral center for glycerol phospholipids or glycerol 3-phosphate. This is a chiral center. So the molecule can either exist in the L form or in the D form. So in the L form, you can say this is glycerol 3-phosphate. That means the phosphate group is attached to the third carbon. For the D form, the phosphate group is attached to the number 1 carbon atom, so it is called D-glycerol 1-phosphate. So in naming such compounds like this, the SN numbering system is actually adopted by biochemists. So we usually put the SN as a prefix before the name of the molecule. So that's why you can see here we have the SN glycerol 3-phosphate. Why is it 3-phosphate? Why not 1-phosphate, or why not 1-phosphate? Because there's an agreement by biochemists that whenever, if you use the deuterium atom to attach to this number 1, this becomes an S configuration, which is called a pro-S configuration. So when it is actually done that way, then the carbon here becomes the number 1 carbon, the chiral center becomes the number 2 carbon, so the other carbon becomes the number 3 carbon. That is why it is called glycerol 3-phosphate. So these are some examples of phospholipids that are found in biological system or biological membrane. We have the phosphatidylcholine. Here we have the fatty acid, but our major emphasis is on the choline. This is the choline. Major emphasis on the choline. So the choline, which you have here, makes this phosphatidylcholine. That's where actually the name is actually gotten from, the choline function, or the choline moiety. Now the choline is an alcohol that contains two carbon atoms bonded to a nitrogen that has 3 methyl group attached with a positive sign. So this is the choline. Here we have the phosphatidylethanolamine. You can see that the major emphasis is on the ethanolamine. These are amine alcohols. They are amine alcohols. So you can see that the esterification is actually at this point because of the phosphate group here. So there's an attachment here. So if we want to draw the structure of the choline, we'll say CH2. CH2 OH CH2 to nitrogen plus, which is further bonded to CH3 CH3. So this is a choline. This is a choline. So when the hydroxyl group is esterified to the phosphate group, we say it's a phosphatidylcholine. For ethanolamine, we have OH to CH2 and NH3 plus. So NH3 plus. So this is an ethanolamine. So it is the hydroxyl group that is esterified to the phosphate group. That is why it is called phosphatidylethanolamine. We have the phosphatidylserine, where the alcohol is an amino acid called serine. So here we have, for serine, we have, let's say hydrogen, we have the COO minus, we have the NH3 plus, then we have the CH2OH. This is serine. This is an amino acid serine. So it is the hydroxyl group at this point that is esterified here. That is why it's called phosphatidylserine. So we have phosphatidylglycerol, where the same glycerol is actually bonded to the phosphate group. So we have the phosphatidylinositol. This is inositol. This is a polyol. We call it a polyol because it actually has so many hydroxyl groups. So we have phosphatidylglycerol, which is cardiolipine. Is the major phospholipid found in the membrane of myocyte. Usually, let's not just say myocyte, but let's look at the heart. It's a phospholipid that is found in heart muscles. So that's why it's called cardiolipin. So here we have two diglycerol, they call it diphosphoglycerol. So we have the first phosphoglycerol or glycerol phospholipid. We have the second glyphospholipid, which is joined together by a glycerol. So that's why it's called diphosphotidoglycerol. So the two individual glycerol phospholipid is joined together by a glycerol backbone. That's why it's called diphosphatidoglycerol. So it is two phosphatidoglycerol joined together by a glycerol backbone. This molecule is called the cardiolipin. So these are major amino. Okay, for example, the ethanolamine, the choline are the major amine alcohol that make up the phospholipids. So you can see you have the phosphatidylcholine, phosphatidylcholine. These are the major membrane lipids, and their relative abundance in different membrane gives the particular membrane its chemical and physical properties. For example, the hepatocytes, the erythrocytes, the different cells of our system, of our body, have different composition of these phospholipids, and it's the composition of these phospholipids that gives the membrane its physiological or chemical and physical properties. So this is a glycerol of the most common phospholipid. You can see here we have the phosphates, we have the glycerol, and we have the fatty acid. Here we have this is a saturated fat, this is unsaturated fatty acid. You can see the bend of the fatty acid here, and this bend actually makes the molecule not to have a good compartment. So it actually contributes to the fluidity of the membrane. So when we say membrane is fluidic, it is the properties of this bend that makes the membrane fluidic, and also the cholesterol composition of the membrane. So in the chemistry and structure of sterols, or look at how cholesterol can actually make the membrane more fluidic. Thank you very much. Do not forget to subscribe to my channel.